All right, we're moving on to DNA and nucleic acids. So I've never actually had my DNA analyzed like this, but you are all going to get a chance to isolate DNA from a piece of fruit and a vegetable and from your own spit or the spit of somebody else in your household if you are not into spitting. And the thing I would tell you is DNA is the red herring. So you've all heard of DNA. So 30 years ago, 30 years ago, I guess, when I was in graduate school, and my aunt, actually, no, I was an undergrad. And I remember sitting at the table with my Aunt Annie, and she said, well, what is DNA? And I was trying to explain to her what DNA was. But now it's like everybody knows what DNA is. In fact, at Christmas, I mentioned something about telomeres. My mom knew like all about telomeres. So we'll get to that. But DNA has become the red herring. When I talked about this last year, I remember my students all looked at me like, what's a red herring? So herring is this fish that's shown there, and they're silver. And if you go to an aquarium, when we used to be able to go to aquariums, and you would stand in the middle of a glass tank that was all the way around you, and it had these silver fish, and they just kept swimming and swimming, and then all of a sudden they would all change direction because of the morphogenic fields. But if one of them had been red, you would have only looked at that. You would not have noticed anything else. And that's what a red herring is. And that's what DNA has become, is we've been so distracted by the thought of it that it is now become the red herring. But more on that. We're going to look at the chemistry first, because that's what I'm required to do. And then I just want you to think of this in a different perspective. So there goes the red herring. And, oh, but first, this is the elephant from our kitchen has moved to many of your computers. Do you have a paper presentation? Now, I'm delighted to say that a couple of you do. I think about 25%, which is actually typical, even when we meet face-to-face. And so many of you are going to be afraid to come to my Zoom office hours because I keep asking you. But please don't be afraid. Please come. Let's figure out over the next, ideally, week. Again, a lot of people change. I've had people change 2 days before the presentation. But we do, and hopefully maybe today's presentation will whet your appetite, give you another thought. I'd hope the neurotransmitter one or hormone one might. The hormone one, I got one bite. So just a couple things. Do you have a topic? And does the topic interest you? So several people have said to me, oh, just assign me a topic. And I have said multiple times, so I'm going to say it again, is you have to pick a topic because you have to be interested in it. And in all honesty, if you are not interested in anything, I'm not sure how I can help you with that. But it has to be something you want to learn more about, something that interests you. And there are millions of topics. So come to an office hour and talk to me. You can say, hey, you can send me an email and say, I want to talk to you privately, and just be the first one there, and I won't let anybody else in or come at the last 10 minutes, and I can kick people out and talk to you one-on-one. But it's not that big a deal. We just need to find something that interests you. And again, over the next week or two, all right, you do need to be able to find studies, scientific studies about it, and those studies need to be ones that you can explain. So you don't have to read a 40-page study and explain it in detail, but it should have, you need a solid paragraph in your words. I don't want big quoted text where you're using words that I can't pronounce, and I'm doubtful, well, maybe you can, but yeah. So find studies and that you can explain in a way that is interesting to you. Not explain to me, but explain to your mom or your dad or to your Aunt Annie. All right, and depending on it, so let's say your topic is morphine. There's lots of history on morphine. Interesting facts, that is so different depending on your topic. So say irisin, somebody picked irisin. The history of irisin, there's not a lot because it was only discovered, I think, in 2012. But you can talk about why it's named and stuff like that. And you do need to state your sources, at least 3 different sources. And you will be doing a presentation over Zoom. There'll probably be about 10 of us in each presentation. I will not be recording them. We usually do a potluck with this, so you can bring your own potluck and eat during that. All right. So we're going to talk about DNA, and usually I'm doing this presentation, the first half of it right after you took the exam, and actually I'm doing it right now before you've taken the exam, but you won't see it until right after the exam. And so this first part are some questions I will ask my kids class, because the lab you're going to be doing is the lab I do with the kids, and it's really fun. It's pretty cool. So DNA is 2 very, very long strands. And the question is, what shape do these 2 strands make? I'm pretty sure you all know this, but some of you might be like Joey, that you are not into biology. Some of you are so into biology and have told me how much you love biology, and that is wonderful. And some of you have not. So I'm going to go through some of the basics. So I would ask the question, does it make a circle? And that's a trick question, because in bacteria, it does actually make a circle. This is from a bacteria. And so they're called plasmids, and they're much smaller than our DNA, but it is the famous 2 strands, which does form a helix. And so there's SpongeBob. It doesn't make a star. It makes the famous double helix. And so there's a famous book and a famous movie about the discovery of the structure. The strands are held together, and so it's like a zipper. And that's actually important because we need a way for it to pull it apart for replication. And I will get to that when we start looking at the chemistry. Now, the gummy bears are not what holds it together. It's going to be hydrogen bonding. So hydrogen bonding, so it's something we can jiggle apart easily. So like a zipper on your jacket. And I do have a challenge that is in the lab that I'll be posting this week. And that is the healthy, you're going to do your regular lab, and then there's a bonus. In the past, we've always made these, which is the 2 strands of licorice with the gummy bears or marshmallows, which is really not healthy. We just spent like a whole month, 2 months of me telling you not to eat sugary processed stuff. And so I Googled, hey, is there a way we can make this healthy? And I had some ideas, and I got pictures just like the one shown there. And so I decided, maybe you guys come up with a really cool idea that's even better than my idea. So it's in the lab that I'll be posting, and let's move on. So how many letters are in this genetic alphabet? Well, there's 4, and we'll be getting to this again when we get to the structure. You don't really need to be writing down anything yet. I should be saying that at the beginning. This is still very hard on me, not having anybody present as I'm doing this, because usually I see you there and I can say, just wait, I'll tell you when you need, this is really important. But AGCT, or ACGT, sorry, when I learned it AGTC, and that's what they stand for. And you'll have this in front of you. It's in the homework set. So I guess we're on study set 9, the picture of each of these, and we'll get to them. So these 4 letters make a very long book. And this book is in every cell of your body. So if you could type 60 words per minute, which is kind of average for some people these days, and you typed all day, all night without a break, how long would it take you? This is how long the code is. That is 3 billion letters. So this would be if you typed, gosh, I don't even remember what the answer is. So it does take more than 9 hours or even 95 hours. It takes more than 95 days. This is nonstop. It would take you 95 years to type your genetic code, just yours. Pretty amazing. And that's in every cell in your body. If you stretched your DNA in one cell, how long would it be? Well, the width of a $20 bill is 2.578 inches. And it's a lot longer than that. Would it be 2 feet? So a foot used to be the length of the emperor's foot. Would it be, so last year when I did this was during the playoffs. and the Portland Trailblazers were playing the night I was doing this. And so Damian Lillard apparently is 6 feet 3 inches, which actually is the answer. It's about 2 meters long. Every cell in your body has DNA. It's about your, well, longer than my arm with my length, my arm span. Probably John, he's pretty tall. 7-ish feet, well, maybe, depending if you're as big as these guys are. But that's every cell. So go Blazers. It's not happening right now. So I think of last year's class. The next year I'm doing this, I'll be giggling thinking of this year's interesting basketball season. So if you put all the DNA molecules in your body end to end, it would go more than across Oregon. It would go more than across the whole United States. It would go more than around the earth. This is just your DNA from every cell in your body. It would make it to the sun and back 600 times, actually over 600 times. Some of you are a little bit taller, probably have more cells. Some of you are bigger, some of you are smaller, but still, that's a lot. And it's all squished somehow in your cell. And if you prefer, you could go around the solar system twice. Yeah, apparently there's something on the other side of the sun that we can never see. It's a darkness. It's really cold. And that's what they were calling Planet X, but they've now decided it's not a planet. We don't know what it is. All right, there is these guys, histones. You do need to know this. This was a question on your test. But histones are proteins. DNA is this long strand. It's double-stranded and it's long, right? It's somehow, we got to take those 2 meters and squish it inside a cell. You can't see a cell with your eyes. You can see it with a microscope, but we got to squish it in. And so this is how it is done. It's wrapped around these guys, which are called histones. So these spools, and it's wrapped around lots of times. And then those spools kind of come together in this conglomerate, and that's how our DNA is. And these histones allow methylation to happen to them, the protein. So a methyl group is a CH3, and so that's a covalent modification. And how the histones are methylated or acetylated, or there's like 20 different ways they can be changed, determines what DNA is expressed or not expressed. And this is what's really cool. Oh, that's a picture of this middle part is the histone, because it's look at all those alpha helices, and that's the DNA, the double helix, being wrapped around it. Right, and I like thinking of my histones being beautiful colors. And that's another picture. So this is how your DNA is all wrapped around the histones and compacted inside every cell. I believe I showed this picture before when I was talking about covalent modification. And it is studies that are starting to come out. You could not find these studies. This is a really hot new field. And so this is just from a couple years ago. And it has to do, so an acetyl group is a 2-carbon group. A methyl group is a 1-carbon group. There's also longer groups that can be added. But depending on what diet you ate, depended on how this was methylated or phosphorylated and determined what genes were expressed. And I call these the guardians of the genome. Oh, here we go. Let's get some ideas. Nobody's picked soy as a topic this year. But these guys are called isoflavones, and these are specific to soy. And so this guy down here is one of the estrogens, which is found in all of our bodies. And these are phytoestrogens. So phyto means plant-based estrogens. And they look similar, and they have an effect on how our body, steroids bind to our DNA and can change the expression of our DNA. These guys also bind our DNA, but they have a very different effect. So you're going to find, this is what I was trying to talk about, you're going to find these studies that get deep into the biology, and that's great if you understand that. Congratulations. It's usually somebody who spent their whole life working on this, but we get the idea. It's going in there to the DNA, and it's changing how our DNA is being expressed. Anyway, one of the things it does is it goes into the DNA and it activates a gene called the tumor suppressor gene, which does exactly what it says. It suppresses tumors. It turns them off. That would be a good thing. So I call them the guardian of the genome. It's like the guardians of the galaxy. These guys are protecting your genome. And methylation, that CH3 actually silences expression. And these guys can do the opposite. They actually will remove the methyl group so we can activate the tumor suppressor gene. And some people pick their topic based on the cartoons. So yeah, it demethylates it and unblocks it. And there's the soy maize. That doesn't work so well. All right, sulforaphane, broccoli, are all the cruciferous. I talked about this before when I talked about enzymes, but sulforaphane is this end product. It is this molecule, which is found actually in all these wonderful foods, downregulates 63 genes in cancer. It's a super duper guardian of the genome. And it increases, so it demethylates the tumor suppressor genes. So we get both these things going on. Yeah. So what's this guy's name again? Anyway, he's saying eat your broccoli. Groot. Groot would say, he just says Groot. So remember, chew your food, because that releases the myrosinase, excuse me, which breaks this down. So this is how it's found normally. This, again, is sensitive to heat. So either chew it if it's raw, chew it even if you cook it. But if you cook it, chop it and wait a little bit, 20-ish minutes is what they say. And yeah, you are what you eat. There is no sulforaphane in this. There's only, this is going to methylate the heck out of your genes, out of your histones. All right, look at that smile. Look at all those endorphins you're releasing from just looking at that smile. All right, you can Google this. Just Google broccoli, the DNA whisperer, on YouTube, and it's a TED Talk, and this guy's very charismatic, and he does a really good job about talking about it, and maybe that will inspire you. It's a really funny talk. It's a really fun talk, and he gets a little bit into the science. All right, coffee or tea. Now I'll tell you, this one is not available, neither coffee nor tea. They actually were two of the first ones picked, so, but they are both. Coffee has all of these amazing chemicals. Remember, this is a chemistry class, so please make sure your topic deals with chemistry. Of course, you will be getting into some physiology because biochemistry and physiology do overlap, but chemistry looks without being afraid of carbons and hydrogens and looks at molecules. So major, these guys also demethylate or do things like that. That's in green tea, EGCG, powerful, powerful antioxidant. Matcha has also been picked, but look at all these other ones, all the berries. Somebody picked strawberries, but it is possible, perhaps, yeah. But these are the big ones. I would actually encourage you. Soy, broccoli, those are available. And spices. Pick a spice of your life, right? Turmeric. There is so much that goes on. We talked about ginger previously. Ginger also might show up on this slide. Turmeric is a powerful guardian of the galaxy. It turns on the guardians. It activates TIMP. I honestly have no idea what TIMP is because it wasn't around my days back in school, but it is tissue inhibitor of metalloproteinases. So a proteinase would be something that's an enzyme, and we are inhibiting them for some reason, and there we go, changes our cell regulation. And so it affects blood pressure. So it actually decreases inflammation of the vessels, and that is a cancer cell exploding. It actually makes brain cancer cells die. They die. All right, this is not turmeric. I'm pretty sure that's saffron. I had somebody do a really awesome paper on saffron. Lots of spices. Basil and holy basil is a different one actually. Stimulates the neural stem cells. And so really big for brain health, Alzheimer protection. And there was the molecule up there, don't forget molecules. And of course, meditation. Regular meditators have an even greater increase in anandamide, in melatonin, in serotonin, and oxytocin, all those things. And there's an epigenetic shift. Also, gratitude. And we're going to be talking about that more. Gratitude apparently causes a change in your genes, what genes are expressed. Epigenetics means beyond genes, beyond genetics, that it's not what genes you have, your DNA sequence, it is which ones are expressed. And yes, your diet affects that, absolutely. Your diet affects your attitude, and it is your attitude probably has the biggest effect. And when you go into a state of gratitude, which meditation, heart-centered, that everything you do is for love, not for yourself, for love of others, there is a change that happens. Service, being of service. That's me. I had to cut the picture down because there were a bunch of kids in the picture, but being of service just to be of service. Every picture, everybody is smiling. And then this is a picture of a gal I sponsor in Nepal. They have been on lockdown for 6 weeks. They continue to be in these countries, the poorest countries in the world. They've been on lockdown. They don't have any cases. They don't have a good medical field, but they're also running out of food. And she had a birthday a week ago. So happy birthday, Shristi. And she sent me this picture. And she's just smiling. And she's so positive in her emails. And she and her friends get together online and study together. And that's what she's doing. She said she's studying so much. They don't know when their exams will be because they don't know when this will end. And then once a week they come out and the lines will be miles long to get food. Anyway, smiling is huge. Attitude. So, do your genes determine your destiny? Or do you? It's a red herring. And I know it's me that determines my destiny. Bruce Lipton. So Dr. Bruce Lipton, amazing. So the study of epigenetics is the science. Science is proving our body's ability to heal and repair itself is greatly affected by our beliefs, thoughts, emotions, and intentions. They have a profound vibrational effect on the continually evolving genetic code. We are the programmers of our code. DNA activation is our software upgrade. Your beliefs, it's not your belief, it's your knowing, it's your attitude. Every picture of him, he is just glowing. And he says he was not like this 20, 30 years ago. He is listed as 100 most influential spiritual people. This guy was electron microscopy, PhD. He did all this research, and then he realized he did these studies, and he took the DNA out of the cells, and the cells did not die. The cells did not reproduce, but they didn't die. They kept going on. And he said, what the heck is going on? And then he did studies with stem cells. And so a stem cell hasn't picked what it's going to be yet. And depending on what environment he put them in, they became different things, muscle cells or this or that, and changed everything for him. So your children's genes reflect only their potential, not their destiny. And as parents, it's up to us to provide the environment, not just for your children, but for your own genes and for all the people that are in your life that allows everyone, because we're all children of each other. So allowing all of us to develop to our highest potential. Things to think about. So if your genes are not in charge, who is? So still believe in the red herring? Let's see. When the protein is needed, it is actually a signal from the environment that activates the expression of the gene. So the gene is your DNA. The gene is a sequence of DNA that codes for a protein, but it is a signal from the environment. That is what epigenetics is. It's not the gene, it's what determines what genes are expressed, and you are in complete control of that. Don't let anyone tell you otherwise. It's like, yeah. All right, here he goes. Goodbye. Bye-bye. So the moment you change your perception is the moment you rewrite the chemistry of your body. All right, but we're going to keep climbing up that life ladder, and let's look at the structure. So we're doing nucleotides. That is our topic. DNA is a nucleotide. We'll get to that fancy word in a moment. So you're going to start taking notes if you haven't been. And a lot of you probably just wrote down all those words. So DNA and RNA, the difference between them. One here is DNA is 2 strands. That's what's shown here on the right. And the RNA is just 1 strand. But they are both made up of nucleotides. Now, again, normally I'm teaching this at the blackboard, and so I should have moved the slide later. I just realized. Too late now. We're going with it. DNA stands for deoxyribonucleic acid, and we're going to draw this out in a minute, the chemical. It is often called the library, the master copy. It is the blueprint of your genetic information. It carries the genes for who you are. Do you have brown eyes or blue eyes? Do you have a hitchhiker thumb? So if you hold your thumb up in front of you, my thumb is a hitchhiker thumb. My thumb actually points backwards. It doesn't point straight up. And that's actually a genetic trait. All right. So are your earlobes attached or not? Yeah. It is found in the nucleus. So the nucleus is you have a second membrane in your cells, and inside that is the library, the nucleus. So we do know DNA is absolutely important. It is the library. It has an extra protection around it. And a nucleus is only found, I spell this with a K because that's how old I am. It was spelled with a K back in those days, and then Americans had to spell it with a C. So there you go. And this just means with a nucleus. And your DNA is pretty much the same in every cell, roughly. There's going to be a mutation, so there might be something off, but it's huge. Oh, sorry. There is only 1 copy per cell. So every cell has 1 copy of DNA, and they have the same copy. Except if you were writing a book that is 3 billion words long, which is really long. I have a slide later, it tells you that. You would make a typo. And so there's probably typos in your cells. We'll get to that. All right, it is 2 strands. Compared to RNA, RNA is ribonucleic acid, and RNA is the middleman. We'll talk about this more in the next slideshow, but it carries the message from the DNA to the site of protein synthesis. And that's what the next slideshow will be about. And so many, one of the places, this is actually my favorite thing, is they call the DNA is the blueprint and the RNA is the working drawings. So you have the blueprint that never actually leaves the architect's office, right? It's the master blueprint. It's the thing that's worth so much. And then you make these sketches that you take that the guys on the scene that are doing all the work, they use the working drawings. Just as important, if not, well, everything's important. It is located in both the nucleus and the cytoplasm. So because DNA is the red herring, there is the nucleus inside a cell. You're either inside the nucleus or you're outside the nucleus. The cytoplasm is anything that is inside the cell but not inside the nucleus. Please do not get that confused with the cytosol. When we get into metabolism next week, I'll have a lovely slide and explain that. Cytoplasm is a word that is only used when you're doing stuff with DNA, because that's the only time you really care about the nucleus. So RNA is found everywhere in the cells, but this is saying DNA is only found in the nucleus. All right, there's many different types of RNA. Again, that's the next slideshow. And every cell, the RNA is different. Some things are going to be the same, some things are different. DNA, it is the same in every cell. And also, whereas DNA, there was only one copy, RNA, there's thousands of copy, thousands, hundreds of thousands, millions. I have no idea. There's numbers that we don't even have numbers for. There's a lot. But it's different in each cell because every cell is different. And it is one strand. The other thing that is not in this slide, I just realized, because again, I usually have the board for it on, is the size. DNA, I already mentioned, was super long, right? It went, well, one cell, it's your arm width. So it's about 2 meters long. It's 3 billion base pairs. RNA, oh, it's anywhere from maybe 10 to 50 to 1,000. So it's short. I mean, 3 billion is like the amount of money Bill Gates has. And you and I have $10 to $1,000. So there's really not a comparison. RNA is much shorter, much shorter. All right. We say a millionfold and everybody's like, okay, a millionfold. And we're not quite getting the degree of that difference. All right. And this is here, me thinking about Joey. I talk about this more in the next slideshow, but just to make sure, because not all of you have a strong biology background, DNA is the blueprint. It's going to code for the RNA, which is the working drawings, so the middleman. And then the RNA is going to have the code for the protein. The protein, they are the workers. They are the ones that are making your cells do everything. All right, and this is ironically called the central dogma. That's our next slideshow. Oh, but we're not done. As you can tell, what this slideshow is about is we're going to look at the structure of nucleic acids. So again, DNA is 2 strands, RNA is 1 strand. But what they have in common, they are both nucleic acids. So we just got done with several weeks about proteins. Now we're doing nucleic acids. And nucleic acids are made up of nucleotides. And that is what these are. And we're going to draw them. So if you don't have your pencil and paper, pots, go and get them. And we're going to do some chemistry. All right. So here we go. How do I play?
Hello and welcome. We are going to draw nucleotides, or a nucleotide, and then we're going to put them together and make a dinucleotide. So nucleotides, if you've been watching the slideshow, it's made up of 3 parts. So over there, what is shown is the base. So this is simply called the base, or the heterocyclic base, or the nitrogen base. And it is because of all the nitrogens in it. And on your study set, and in the slideshow, I show a picture of it. So in your study set, and on the quiz, and on your midterm, you'll always have the pictures. You can also Google them. So that's one part. Second part is there's always going to be a sugar. So we're going to have a base, and we're going to have a sugar, and the third part is a phosphate. So base, sugar, and phosphate are the 3 parts. So we're going to start with the sugar, and the sugar is going to be ribose. So what do you remember about ribose from the first week when we were here? Nobody ever remembers very much. We are going to draw pentagons. So over this last month of class, you're going to get good at drawing these pentagons, because not only is ribose going to be important for making DNA and RNA, these nucleotides are also going to be our energy molecules, our ATP, GTP, CTP, and UTP. All right, so you draw your pentagon with the oxygen there, and then we want a number, because the numbers are important to chemists. So 1, 2, 3, 4, and it's a pentagon, so where's number 5? Do you remember? Hopefully you've already drawn it. Number 5 is going to be up above. Now I'm going to draw the OH as a hoe on this side. That will make it easier, and that is number 5. Now every carbon of the sugar is important, and something happens at every carbon, so we're going to go through that. So 1, 2, 3, 4, and 5. So let's go ahead and start number 1. Number 1 is the base. So we can move this over. And there are 5 different bases. 4 are found in DNA and 4 are found in RNA. So DNA has A, C, G, and T. And those of you who've taken biology classes, you probably know that. My background is actually as a molecular biologist, but there was no such thing when I got my degree. So I'm a biochemist. RNA is A, C, G, and U. And if you look at the picture, the difference between thymine and uracil is just one methyl group. So one difference, DNA has the T and RNA has U, which is uracil. And since you guys have your notes, it's easy. You can always look that up. All right, this, at number 1, this was always going to be in the beta position like that. So remember we had an OH at every place. 2 and 3 are going to point down. So that's finishing our sugar. So every position on the sugar. And number 4, remember, its oxygen is what made the ring. So to add the base, we're going to add cytosine, is that H that's sticking down. So here in cytosine, that H, or in guanine and adenine, the H that's sticking down is the H that's going to come off, as well as that OH is going to come off. So in our body's made very brilliantly. It's based off of water. Water's just perfect, beautiful, vibrating molecule. It's a perfect frequency, the shape of it. So that OH comes off, and this H is going to come off. And we're going to directly connect to nitrogen, like that. And the nitrogen comes along with the whole package. So you will draw. Now, there are 2 different types. There are the purines, which have a 2-ring structure, and the pyrimidines. I picked one that was pretty simple. My H back on there. And my double bonded oxygen. All right. Now, to tell you the truth, the picture that's in your homework right now, at the time of my filming, they have them all flipped as if we were always drawing your sugar over on that side. I don't know why, and ends up confusing students. I don't really care which way you flip it, but I do know that this double bonded O is going to point that way, and it will help me explain a later step, but it's not that big a deal. So there's the base at number 1. Number 2 is also important, because what happens at number 2, you're either going to have an OH, which makes it ribose, the sugar, or you're going to have just an H, so it's halloumi, and that is deoxyribose. So if we lose the oxygen and just put a hydrogen there, we have DNA. We're making a DNA nucleotide. This is how, looking at number 2, you're going to know if it's a DNA or an RNA. I keep twisting. All right. We'll go ahead. It doesn't matter since we have cytosine, which one we do. All right. Number 3 is going to connect to the next nucleotide, or race sugar. And we'll do that in a minute. So this is going to connect to the next. I just say NT for short. All right. Number 4. Oh, that's what made that pentagon. So that was our pentagon. So it already did its thing, so that we could do all the rest. So number 5, well, we have one piece left, and that's the phosphate. So number 5 is the phosphate. And that's why I like drawing it on this side, and you will too. So a phosphate, you're not just going to write PO4. You're going to take the H off, and you're going to put the P, and this P has 4 oxygens around it. One of them is a double bond, and the others are just oxygens. Now, if the oxygen is just hanging out there without a second bond, it is ionized. A lot of books still show it with a hydrogen, but that's a mistake. If you remember, you just had a midterm about this with amino acids, our body ionizes things when it can. And so that phosphate, those hydrogens come off. Now this one has a double bond, so that oxygen has its octet, and this oxygen has a single here, and then there would have been a single there. All right, this is going to make it very special about how DNA ends up making its structure. All right, so at number 1 we have the base connected by the nitrogen. At number 2, right now we have a deoxyribose, so DNA. Number 3, we're going to do our connection, and I just realized I didn't give myself much room, but hopefully you guys have a whole piece of paper, and I should have told you that when we started. All right, and then number 5, we make our phosphate. So let's go ahead and add the next one. So the H is going to come off. And this oxygen is going to hook up to a phosphate from the next nucleotide. Now, only one of the oxygens now has a negative charge. One of them is hooked up to the oxygen at number 3. One of them hooked to the number 5 carbon. It's going to go down to our next pentagon. So there's 4, 3, 2, 1, and one of them is a double bond. And right at number 3, you can put an OH, but in all honesty, it would go to a next phosphate. In DNA, we're talking 3 billion. It's huge. It's really long. At number 2, it is just an H. If that one was an H, this has to stay as an H. If it was an OH, it would stay as an OH. And then we would put our next nucleotide. And if you want, we'll make it a guanine. So make your guanine, and let's see if I can find my eraser. There we go. So that's what makes this a chemistry class, is we draw the structures. And this goes back, we keep coming back to those carbohydrates, that first thing that we drew. So we have our huge oversized pentagon there. Number 1 has a base down here. Number 1 has a base, just a different base. Number 2 is our OH. Number 3 is hooking up to the phosphate for the next sugar, for the next nucleotide. Number 4 made our pentagon, and number 5 has a phosphate. Now, we could keep going and going and going. I ran out of room, so I'm going to stop. But we always read DNA from the 5' to the 3'. And this is what's known as the 5' end. So the end that has that free phosphate, it should be looking a lot like when we did the N to the C terminus, because it has, that's where we're going with this. All right, this, if we kept going for 3 billion, can you imagine? That would take us like the rest of the year. This whole pandemic would probably be over and we would have moved on. We would be in a great meditative state if you just drew 3 billion at least. All right, the 3' end would be the one that is not hooked up to anything else. So we always read 5' to 3'. All right, and then the order that the basis is in is how we would read it. So ours right now is just CG. It would, of course, keep going and going and going and going, and we'll do practice with that in a minute. The other thing I want to show you, while I have this here, is something that is why Watson and Crick deserve a lot of credit. So the story is that, so Watson was an American, young, like 23 or 25, and had gotten a free fellowship to go and study. He had made this wonderful PhD on, I think, bacteriophages. And they put him in a lab with Crick, who was in his 30s and still didn't have his doctorate because he was perfectly content to just be in the lab. And the two of them got along brilliantly, and they just talked about what was the possibility that everything about us, the color of our eyes, had to be explained by some molecule in our body, and the push was on. So this is right after World War II. England had, and the United States had bombed a good part of the world, and people in England had been bombed so much that they wanted to study life. And so there was all these people who would come into this field to study life. And there was this beautiful woman named Rosalind Franklin who did these beautiful X-ray crystallography and had figured out, well, it must be a helix. But these guys, Watson had a photographic memory and saw a picture of it, and he said, well, it is a helix, and he explained everything to Crick, and Crick made this brilliant assumption, and he said, okay, well, if it's a helix, it's two-stranded, not like the one-stranded one that had won a Nobel Prize for Linus Pauling, the alpha helix. This is going to be two strands, but he said the two strands are going to run opposite. So while this one's running this way, the other one's going to start 5' here and go 3' here. Now, if we were in class, this is where I make you all turn your paper upside down. I stand on my head. I'm not going to do that for the video, because if I fell. But I'll try and draw my pentagons upside down. But basically, this is going to go upside down. So we'll see if I can even draw my CH2s upside down. My oxygen and my phosphate. You guys should all just try and draw upside down, because it's really fun. All right, this is going to be my oxygen to my next P. Oh, good for the brain. This is just an H. Now, this was guanine. It's going to hook up to cytosine, and this will keep going on, right? This would hook up to my next pentagon. I have to think how to draw a pentagon upside down. But it's a pentagon, so there's not really a right side up or upside down. So this is going to be cytosine, and our cytosine, I'll draw the N upside down. Is that right? That's what I wanted to show you. Is double bond with O down here, and something like that. All right, and we keep going. What happens is the G and the C make hydrogen bonds. So look at that. There's an NH. Do you remember NOS? Whenever you have a hydrogen attached to an NO or F, NOS, the little G there, it's the hat that the gnomes wear. I don't have a hat. Anyway, we're going to see this hydrogen that's attached to the N is going to be attracted to that oxygen. And this hydrogen is going to be attracted over here to this nitrogen. Anyway, you get these attractions. So these are hydrogen bonds, or H bonds, between the nucleotides, between the bases. So G, that's why I always show dots, G always bases to C, and A always bases to T. So there we go. All right, and with that, oh, we're going to do one more thing. This is on your first study set. So that's the idea. I will have you draw a nucleotide and hook another one on, and I often make it bonus if you want to try and flip your paper and draw the next two, and you would continue. What we're going to do is we're going to do one more piece with this, and we're going to go ahead and just write out a sequence. So we'll say 5, and I'll just say GCTAGC. All right? So 5' to 3'. And we're now going to make the complement. So the complement is going to be the other direction. So these are called complements. Like, you look really nice today. Oh, your hair. Dominique, your hair looks extra special today. Right? Complimenting. Did you know gratitude makes your DNA longer, the telomeres? We'll get to that. All right. So anywhere you have a G, it's going to complement to C. Anywhere you have a C, so my GCT is now a CGA, except I'm reading backwards, technically. So T, C, and G. So C always goes to G, A always goes to T. These are called base pairs. And these are complementary strands. So DNA has 2 strands, and they always complement. And I'll see you in a few moments.
All right.
Hello.
Oops. So a review, you would number, so these are the sugars, either ribose or deoxyribose, and it's here at number 2, ribose has the OH and deoxyribose has the hydrogen. And a reminder, number 4, there is just a hydrogen because it's OH made the ring, and then up above it would be number 5. And number 1, we're going to pull that OH off and we're going to attach the nitrogenous base. So there we go, ATCG. So purines, caffeine and chocolate have purines in them. And adenosine is the one that caffeine mimics. Adenine is called adenine, and once it's attached to the sugar, it's called adenosine. And so attached to the sugar, it acts also as a neurotransmitter that helps us to feel drowsy and to fall asleep, so that melatonin can be released. So is it the melatonin that makes you fall asleep or the adenosine? Anyway, and then there's guanine. And so the guanine is going to H-bond to the cytosine. The adenine is going to H-bond to the thymine if it's DNA, to the uracil if it's RNA. So thymine, cytosine, and uracil are pyrimidines. All right. You just need to know A always goes to T. G always goes to C. If we're talking about RNA, the U just replaces the T. Oh, and actually something I said I would point out, and this probably has to do with the thing I went on about, the histones are methylated, that the thymine, which is found in DNA, has this methyl group. So is it always there? All right, nucleotides are made up of these 3 pieces. It's probably going to be a question in your multiple choice. So you have the sugar, you have the base, and you have the phosphate. All of this is covalent bonds. This is called deoxyadenosine monophosphate, or short DAMP. The D means deoxy, that we have just an H here. The A means it's adenosine. You'll always, well, it's open book, so you have that picture that's in your homework set. M means monophosphate. So ATP, the T means triphosphate, and there is no little D in front of it. So if it just says ATP, it would mean there's an OH here. So it's actually ribose. All right, and I feel like I double clicked. Nope. So there you go. That's the backbone. We call it the backbone, is sugar, phosphate, sugar, or phosphate, sugar, phosphate, sugar, phosphate, sugar. And we call this the 5' end because the phosphate would have nothing on it. And this down here is the 3' end because there is nothing else. The bases are sticking out and they're going to hydrogen bond when we put the other strand attached to it. So this is a covalent bond, backbone. That is important. You can't shake a covalent bond apart. And so here we go, 5' end to 3'. This is the sequence, TGCA for this one. We always read 5 to 3. And that is the primary structure, just like we had with proteins, is the sequence, always read 5 to 3. So TGCA is not the same thing as ACGT. Yeah, you're dyslexic, this could be a nightmare, right? That's just a picture of the very first DNA sequence that was determined, which was insulin back in the 50s. And this was by Sanger, who won a Nobel Prize. First protein that was sequenced, and then first DNA that was sequenced. And so you can tell this was a typewriter that typed it. So it's really impressive that they were able to do this. Now everything's done, of course, with our computers and figured out. This is key, that the two strands are held together by hydrogen bonds. So it is a zipper that holds it together. So your coat, it's the sleeve, you would have to rip it. So to cause a backbone break, the ribbon here, you would have to rip the sleeve off your coat. But it's just a simple zipper, opens and closes the two strands, which implies a really nice way for replication, which was a very elegant idea that James Watson had. So there's two strands, they run opposite directions, and the outside, the backbone, is the sugar phosphate or phosphate sugar, and the inside is the bases or nitrogenous bases, and H bonding is what holds it together, and always A to T, G to C, always. And it's a double helix. And there you go. You can make wonderful cartoons. All right, that's just a picture of all these famous people. Nobel Prize, when they made it, you can only have 3 people on a Nobel Prize. It's a rule. There have been a lot of people who should have won a Nobel Prize, but they don't allow 4. It doesn't mean it's a good rule. And if you're dead, you can't get a Nobel Prize. Rosalind Franklin had already died when the Nobel Prize was awarded for the DNA structure. She died from cancer. She was the one who was she was the person who did all the x-rays. She was brilliant. And she's probably more famous because she wasn't on the Nobel Prize. She died from probably the exposure to the x-rays. She had breast cancer. And I honestly, it's a prize. It's, yeah. So there's all this controversy and stuff about it. Let's all get over it and move on. This is how they had to make it. There was no computers. These guys built it from scratch. They did none of the lab work. She did the x-rays. It was Maurice Wilkins' lab. He did a lot of the initial studies that figured out how to be able to make the x-rays. He actually is a physicist who helped in the developing of the atomic bomb, and he didn't want to be in the death field, as I said. And you can get bobbleheads, you can get earrings, you can get all sorts of wonderful DNA paraphernalia. All right, base pairing, already talked about. That's what I was trying to show you. You don't need to, you just need to know it's H bonding. That would be a question. All right, let's talk about DNA replication. So DNA has to have a way to make a copy of itself, and that is called replication. A couple things about it, and this is more, you get more detail in biology classes, and at one time this was fascinating to me, but every time the cell divides, it makes a replication of the DNA. That's all the DNA is needed. The cell can survive without it. It cannot replicate without it. Both strands replicate at the same time in opposite directions. So that's what this is trying to show. It's happening at multiple places at one time, because if you guys were trying to write down 3 billion letters, you couldn't do it in a couple hours. So you need a bunch of people doing this. And so that's all that's saying. And it is something that was called semi-conservative. So it's hard to see, but one of the strands is this original like light blue tealish color, and one strand is the darker blue new. So conservative would be if one of the new DNAs was all the old DNA and one was all new, but it's semi-conservative. So the two DNAs, one is old and one is new. I don't know. And every time, it's pretty amazing actually. This is a really oversimplification of the basic steps in DNA replication, but there is something called a helicase that unzips the 2 strands, ends with ACE, so that is an enzyme. And then this is a step in pink that is fascinating to me. This step does not use an enzyme. The nucleotides complement. The A knows, hey, there's a T. This is a magnetic attraction. Yes, we talk about it as H bonds. It happens immediately. We could not measure the time. Some things really cannot be measured. I know we've like over scientificized everything. I don't know what the word would be. The A here is coming along with its sugar and phosphate. That happens. Boom. That is the whole key, that the complements happen. And then a polymerase, an enzyme comes along and has to hook up the sugar and phosphate to the next sugar phosphate. Because it's a covalent bond, it requires an enzyme. And then part of this DNA complex, there is a proofreader looking for mistakes, because there are mistakes. There are actually a lot of mistakes that happen. And so it cuts out the mistake and allows the correct one to come in. The mistakes are usually, and I'd have to go back, but I'm not going to. Thymine sometimes attracts G because, so it's always a pyrimidine to a purine. And yeah, there's lots of fun. See, now you're going to understand all the fun cartoons. There's so many fun cartoons in biochemistry. But this is cool, mutations. Mutations are an error or a mistake in the DNA sequence. There's two ways it can happen. One is what I just implied. So which mutant are you? Because we're all a mutant. Did you ever think about that when you watched X-Men? Which one would you be? All right, so spontaneous just means it happens. It happens in replication. As we get older, it happens more frequently as the machinery gets older. I don't know if you can call that spontaneous, because how you age does determine how many more. There are mutagens, things that cause a mistake to happen, a break to happen, things like that. X-rays. So as I mentioned, Rosalind Franklin was exposed to high amounts of X-rays. Smoking, right? We all know that causes lung cancer. A lot of chemicals. So this is benzene. Benzene is flat, and benzene fits perfectly into the nooks and crannies of DNA. It is the classic carcinogen. So when it is a chemical, it's called a chemical carcinogen. We know it causes cancer. Oh, viruses can too. So HPV causes cervical cancer, and sunlight. Yeah, that's the whole thing. But vitamin D deficiency is linked to poor response, poor recovery from the coronavirus. There's so many studies that have been coming out in the past month. Nobody has picked vitamin D as a topic, and you could really have fun with that. So people with really low vitamin D are the ones who are not doing as well with the coronavirus. They actually have known that from the beginning, but all right, air, breathing. We all have to breathe. Oxygen. We're going to talk about oxygen as a mutant when we get to metabolism, because that's a big part of it. And of course, we all know this. If you didn't, hopefully you're not still eating like this. You should just look at this. And if you have Twinkies, we should have done this. Leave it out on the counter. The ants, I don't think the ants would go for it. Sadly, my puppy would. But there's some experiment where some guy has left a Twinkie in his office for like several decades now. Nothing has happened. There's no mold on it. To tell you something, when mold won't grow on this stuff, right? Oh, and that. Yeah, like everybody's like, oh, I know this is not good for you. Oh, and that. Yeah. Oh, yeah, I know it's not good for me, but I just have it once a week. I don't think so. Right? They liken eating French fries to smoking cigarettes. Oh, and that is an attitude. Your attitude is a mutagen. Smiling is not a mutagen. Smiling is the epigenetic shift, the way we want it. I don't see broccoli on here. I wonder why. That was in the previous stuff. All right, so your DNA has a repair mechanism, as I already mentioned, or you can help your DNA repair. So yes, that's me. I made myself really small. That's my house. That's Joey's room. He lives there. All right. So berries are antioxidants. Look at all those amazing things in different berries. Anthocyanidine, sorry. That's the purple ones in blackberries. So many cool stuff about them. Again, strawberries have been picked, but they're filled with polyphenols and anthocyanins, which are amazing. Kiwi is a cool topic. Somebody did that. 3 per day, 60% decrease in DNA damage. That's pretty amazing. And I eat the whole kiwi, including the skin, and that grosses my students out. And I don't get to see all of you make faces at me. But yeah, everybody's like, ooh, the skin's gross. But then they all sit there and Google, and it turns out, yeah, the skin is where all the stuff, there's a lot of nutrients in the skin. But nobody ever taught me how to eat a kiwi, so I didn't know you couldn't eat the skin. Right, loaded with vitamin C. So not only do you see a decrease in DNA damage, you also see this increase in DNA repair. All right, oh, the carotenoids. So you don't want to do beta carotene, you would need to do the carotenoids. There's a whole bunch of them. Beta means it was the second one, and that's part of the problem. So beta carotene is famous because it's a precursor to vitamin A, which we obsessed about these like 8 vitamins at one point, and you need actually all the carotenoids. And it's not just carrots, it's all the orange, red, and yellow vegetables. 5 per day for 2 weeks. There it is. There's beta carotene, gets cut in half, and you have vitamin A. Really good for eye health. And it repairs existing damage. 5 servings per day or 5 carrots per day. Well, that's easy to do. And these are like picked straight out of the ground. They're like the sweetest thing there is. Carrots are amazing. All right, lycopene is any of the reds. Well, not any. I don't know. Is there other reds out there? So watermelon, guava, grapefruit, tomatoes, to name a few. And there's a picture of lycopene because this is chemistry. So you want to talk about the chemistry, the biochemistry, the molecules when you look at this. So it protects from sun damage before exposure. So if you eat guava or grapefruit and then go out in the sun, you actually have sun protection. And then you can get some sun exposure before you have to put your sunscreen on. Because once you put the sunscreen on, you can't make vitamin D. All right, lots of ideas there. Oh, and this was a book I read last year when I was making the slideshows. And then I gave it to one of my students. But there's a thing about oysters. And so if you're really into oysters, if you always go oyster, the digging and stuff, yeah. Anyway, they do something in there too, and I can't remember what now. All right, I don't like oysters, so sorry. But you can do it as a topic. That won't affect me at all. Chromosomes is how the DNA is organized for cell replication. So normally the DNA is just a tangled mess. And now it's organized. It's kind of like when I braid my hair, it's organized. And when it's not, it's like this hot mess. When it's a hot mess, it's called chromatin. And then it just organizes. This is something they don't fully understand, but it makes this. The center is called the centromere, and there you go. And this is, right, this is what's fascinating to me. We have 23. We have an odd number, and I don't know why Rye has 7, but everything else has an even number. I'm pretty sure every other animal has an even number. We have an odd number. It's fascinating. There's other things that are fascinating about our DNA. I can point you in that direction if you want to really go down a rabbit hole. All right. So the Human Genome Project was our National Institute of Health from several different countries, including China and several European, and then some private labs, this one specific. And they decided they were going to sequence. They actually, nobody's ever sequenced our whole DNA, but they claimed in the year 2000 we did it. And they thought we were going to have like 200,000 genes. They thought we were so complex because we're humans. And this number of genes that we have keeps decreasing, which is really interesting because if they claim that 20 years ago they finished sequencing it, how comes we keep having fewer and fewer genes? And it has to do with alternate splicing and stuff. So a nematode, which is this little worm-like creature that I'm not sure you can see with your eyes, but it lives in the soil and makes the soil amazing, has 19,000 genes. We have pretty much the same number. Don't get too big for your britches. But we code for, yeah, something like that. It's an ish. How could you count, really? All right. If you took our 2.9 billion base pairs and just typed it ACGT, blah, 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 it would fill that many pages of the New York Times. That's a really big newspaper. If you haven't had a whoa moment, every cell has that same sequence. You think you could type it in your lifetime? You would be really busy. This used to be me, right? Reading, right? All right, this is the part that is amazing. This is only true for humans. 97%, more than 97% does not code for proteins. So everything that's the red herring is we figured out the code. We can make proteins. That's only like a little bit over 2% of the DNA. Most of it, they say, oh, it's just junk. Oh, it is not. 50% is repetitive. They never actually sequenced it. There's some really interesting stuff here. As they're starting to sequence it, it actually has a vibration to it. It has a sound. And the ends are called telomeres. I'm going to get to that in a moment. This means fasting, as in not eating, actually makes your telomeres longer. And I'm going to talk about those in a minute. There should be another E there. The centromeres were the center part, stability. I don't know as much about them. But 99.9% of your DNA and my DNA is the same. The same. Because although I can't see you, and a lot of you, I don't even know what you look like because you don't turn your cameras on and smile and allow me to have endorphins. But anyway, yes, I have a pet peeve about that. We all have two eyes in the middle, right, on each side of our nose. And there's a third eye between our eyebrows, whatever. And we have ears on each side of our head. And we have a certain number of vertebrae. And we have one stomach, right? And so this was, Bill Clinton was still president in 2000. It was called Our Common Humanity. And yeah, 40% of our DNA is actually the same as a cabbage. This is where you can play It's a Small World After All. You ever been to Disney? Gone on those little boats? So I took Joey when we went, because I'm like, you have to go on this ride. And then they had to stop the boat to let somebody on, and we were halfway through, and you wait, and it does get a little annoying. But in this country, not in any other country that has a Disney, but only in the United States, they actually shut it down about 10 years ago for several months because they had to dredge it, meaning they had to make it deeper. The American boats were bottoming out, and It's a Small World After All. So we have a deeper moat for the boats, because Americans got too big for the boats and they were bottoming out in this ride. What's that tell you? Is that in our DNA? Because only 2% is the proteins. All right, and there you go, me, Mr. Cabbage Head. All right, and this is the red herring. Everybody's like, oh, we figured out what gene's on what chromosome. Good for you. Oh, let's not forget about the microbiome. 53% of our cells, but something like 99% of the DNA in our body is these guys. Let's have some respect for them, eat healthy for them, and for you. So let's talk about telomeres. So telomeres, or telomeres, I've heard both ways. They are described by everyone now as the ends of your shoelaces have eaglets, I think is what they're called, those little pieces that keep your shoelace from fraying. Well, every time your DNA replicates, the DNA polymerase cannot copy the ends. It falls off before it can copy the ends. So you lose, let's say 10 or 15 base pairs at the end of the DNA every time the cell replicates. And so eventually you lose enough that if you lose your telomeres, which don't code for anything, they're just this repetitive end, that the shoelace is going to fray and it all comes apart. And your DNA, you don't want it to fray. That would not be good. So they did this estimate back in the 50s. They've known about telomeres. And it was somebody did statistical analysis and determined that a human can live 82 years based on how long our telomeres are. The thing was, this analysis did not take into the idea, it turns out you can make your DNA longer. Yes, your DNA is not fixed. You can determine how it's expressed. You can change your DNA. Oh my gosh, did I just say you can change your DNA? And yes, meditation is one of the things, but not the only one. Coffee and tea, both, increase the length of your telomeres. Not when you add crap to it, but black coffee or tea, doesn't really matter the type of tea. You can add spices. Spices are amazing. So there you go. Enjoy with a smile, because smiling absolutely increases your telomeres. Exercising with a friend especially, and with a smile, and with your puppy. Yes, because puppies always make you smile. Look how happy they are. I want to know how this ended, because I can't imagine it ending well. Oh, and well, feeding the squirrels, perhaps, because you're being kind and generous. But eating nuts, your diet, nuts are something that increase telomeres, all those healthy things we've talked about. And being playful, letting your inner child out. Being playful is huge. So is it nature or nurture? So my doctorate is in biochemistry. It was in genetics. I worked on genes. I was a gene jock. And I thought it was all nature. It was your genes determined who you were. There was nothing you could do. Oh my gosh. You know, my mom, when she had breast cancer, oh, should I get tested? I was there to see if she had the gene. And it was this whole thing. And they sit there and they counsel you with so much compassion. And all right, it's their belief system, right? Their BS, their belief system. If that is what you believe, then that's how it's going to be. And if you keep eating this, then you are right. You're going to get cancer. Your telomeres actually shrink about 15 base pairs per year. If you eat this, you can actually shrink them faster. So I just previously showed you a slide that could make them longer. Anybody want to guess the #1 shrinker of telomeres? You can all like type in your answers. This is where we should do a live stream and we can see people's answers. And it is bacon. Is anybody still eating bacon out there? Let's be honest here. Let's really be honest here. Are you still eating processed meat? Because you're doubling how fast you're shrinking your telomeres. You're not surprised, you know that. Oh, and the sugary drinks. You might as well just take up smoking while you're at it. Yeah, sugary drinks and smoking decrease them the same. Just a little bit slower. Processed meat is the best at it. And you know what? If it's processed turkey bacon, it's still processed. So please don't ask me, but I have turkey bacon. Really? It's processed. As is the processed tofu crap that's out there right now. So yes, Elliot, absolutely, you are right. If it's processed, it's crap, but processed meat is the worst. All right, saturated fat also was really good at this. Oh, can we have bacon without saturated fat? I don't think so. 1,442 genes are methylated. Methylation means silenced. That is a lot of genes that are being silenced. Genes that would turn off cancer. Your body doesn't want to have cancer. It's kind of like we put a face mask over all these genes. 28 of them are in your adipose. Does it wonder? what's happened to Americans. All right, let's look at a study that came out of the West China Teaching Hospital. They looked at 4 diets. The first diet was called the whole food plant-based diet. Again, not vegan, whole food, plant-based. So again, Twizzlers are vegan, but they are not whole food, nor are they plant-based. So that was diet number 1, the gorilla diet. If you ate like a gorilla, you would be a lot healthier. Diet number 2 was called the macho diet, meaning the American diet. Yeah. Diet number 3 was the Asian American diet, which was this. People go to those Asian American places and say, oh, look, I'm eating healthy. This, oh my gosh, look at the oil dripping off of that. Look at it. And it's all sugar. So most of the Thai places in this country, that's not Thai food. It's pure sugar and oil and salt. Oh, and then this is sad. This was the school lunch diet of Americans. They called it the high energy density, meaning it was sugar, wheat, and meat, highly processed. Anybody want to guess which one had the longest telomere length? They can actually measure this on humans. I don't know how this is ethical. Oh, but wait, how many of you know people who eat like this? How many of you have eaten like this? Like when you were in high school and that's what they served. And you said, oh, I'm gonna go for this today because it's healthier. Really? How many of you are eating like a gorilla? Whole food, plant-based? The only one that increased the length of the telomeres. And I showed you these pictures. These are some of the original pictures, actually. This was the American family. This was Australia. The one from 20 years ago, there is no vegetables on there. It was all meat. It's in my office. And then these are the third world, the poor countries that right now can't go out and get food. But I want to go to their house. I want to go to their potluck. Look at that. There's quite a difference. So what increases the telomeres? Your diet. Look at that. Yum. Exercise. But being playful with your exercise. I go for a bike ride. I tell this story all the time. And Joey was little and I had that, we had an extension and he was pedaling too, but his chain was not attached to mine. So he could pedal backwards. And so we're on the spring water and so we're pedaling. It was the first nice day in April and we'd had rain and rain and rain and it was just packed. And everybody's in their spandex and serious and going, and I'm really serious. And I passed this Asian guy, and he's just biking like the frog, but with no hands. And his feet were probably on the pedals. And yeah, and he was just so happy. He just had his face up to the sun and he was so happy. And I kept pedaling and I made it to our turnaround point before I had to be at home to take a shower to get to school. And I turned back and I pedal again and I pass him. I kind of stopped pedaling for a moment and I looked at him and Joey's just there. And people would yell to me, hey, he's pedaling backwards, meaning my kid, Joey. And I got home and I took that stupid speedometer off my bike and I threw it in the garbage. I've never had one since. And I'll tell you what, yesterday, I was out pedaling. This happens every day I'm out there because it was sunny yesterday. And all these people passed me. And they're all like going to kind of look at me because I'm like biking out there past Barlow High School. And I'm like biking with no hands. I'm looking at the fields. I'm looking at Mount Hood. And I'm just delighted. I have no idea how far I'm biking. In fact, I had to take a detour because the police shut off the street because there'd been an accident. And just enjoy. So who got more exercise? Sleep in the dark. Turn that Wi-Fi off. You got to do it. It will make such a difference. And laugh. Laugh more. Be playful. Be playful. Pick your topic. Be playful. And the number one increaser of telomeres was gratitude. And I'm in a dilemma of what lab 8's going to be. I think it may say mystery lab. I don't know what it says. I'm debating about being a gratitude lab. Joey's joking about this because there's a dead bush in our front of our house. It's been there for 20 years. And so it was time to go, and Joey has to dig it up. So he said he's going to wait until I make this lab. So that will be his service and gratitude for me. And that's just to remind you, service also. But we'll see. If you have an opinion, you can send me an email or tell me in office hours. And all right, just laugh, be playful. Isn't that an awesome picture? And we're gonna stop this recording and have a beautiful day. Smile, be playful. Bye.